I'd like to thank you. It's actually a great honor to participate in a symposium for Gobind. As you know, he was my mentor. I was actually his second graduate student after a gap of 15 years. So it really took him a long time to decide that he actually wanted to have graduate students again, despite the fact that his first graduate student was highly accomplished. In fact, almost intimidatingly accomplished, since he synthesized acetyl-CoA. So today, as Partha mentioned, I'd actually like to talk about three things. The first is my experience as a student in Gobind's lab, and actually the first problem that I ended up working on. The second thing is just briefly mention the cancer work. It's obviously so complicated that I decided that really what I want to do is tell you one story that relates to this pandemic that we've all been dealing with over the past 3 years. So Gobind's philosophy, and the one that I've used in my title, and something that he constantly told me, is that I only work on big problems. When I joined his lab, this was around the time that, as he said, in middle age, he made a turnaround in his interests that he felt turned out to be good for him in many ways. And you've heard from Tom kind of the history of his work. And in particular, you've heard a lot about total synthesis of the gene for alanine tRNA and tyrosine suppressor tRNAs from 1961 to 1967. But right after that, he got very interested in membranes. And his first foray into the field of membranes is to look at the structure of lipopolysaccharides from gram-negative bacteria. And this was between 1972 and 1982. And this was the time when I joined the lab. So I joined the lab at a very unusual transitional time for Gobind, after which, and in fact, overlapping some of that time, he worked on membrane structure and then moved really to his final projects, which were bacterial rhodopsin and mammalian photoreceptor rhodopsin signal transduction. So Gobind, like Dieter, basically would grab a problem and stay with it for a long time. But as you see, he kind of transitioned from one problem to another. But I caught him kind of during this middle point, which maybe caused me to be looking at multiple problems during my career. And you'll get a feel for that. So Gobind always said, again, there's breadth and depth in every scientific field of investigation, and science is pursued in a variety of styles. So I just want to give you a flavor for what my graduate project was like. So basically, as I mentioned, he was interested in the structure of bacterial lipid A. And the strategy that he used, which is really the strategy that he used really throughout his career, is to apply tools and expertise from one discipline to unresolved problems of another discipline. Why did he want to look at the structure of lipid A? The answer is that he knew a lot about phosphodiester linkages in polynucleotides, and he thought that actually lipopolysaccharides were composed of diglucosamine residues that were linked by phosphodiester bridges. So he thought, this is a trivial problem. I can do this. It'll get me into membranes. And so he launched it with 2 postdocs before I joined the lab. But there was a problem. The problem was that lipid A, even though it had this diglucosamine core, that core was derivatized with N and O-linked fatty acids. And it's easy to get rid of O-linked fatty acids chemically, but N-linked are very difficult. And by the time you remove them, you've destroyed your molecule. So Gobind had a solution for me. And by the way, this was my summer project. And that is that slime molds have amidases that feed on gram-negative bacteria. And so I should purify these amidases and then gently break down this molecule. So on this slide, this is a restatement of the problem. And this did turn into my thesis, which is I had an unknown substrate, lipid A. I had a bunch of unknown enzymes, these amidases from slime mold that I had to isolate. It was going to give me a bunch of unknown products because, of course, I didn't know what I was starting with. And from that, I was supposed to figure out the structure of the original molecule. So this is kind of a classic Gobind problem. And to tackle it, one had to be rigorous scientifically, had to have a lot of intellectual integrity, and most importantly, be interdisciplinary. And that certainly has been a characteristic of my entire scientific career. And so in the next slide, I'm still here, so clearly I passed. I actually figured it out. I had a 3-page centerpiece of all the molecules that came out of that project. And I would say that I learned a few lessons. One is try anything. 2 is be fearless. And 3, which is so important to this day for me, and this is really important for any student, is never give up. Perseverance. And I'm going to end with a COVID story, and that is definitely a perseverance story. So next slide. I just want to spend a couple minutes talking about what it was like to work in the Khorana lab. And this is one of the examples of our group photos. It was a very large group. I'm sitting on the lower left. And the other 2 people highlighted were postdocs who worked on my project but never got any papers until I finished my thesis. And you can see Gobind standing right in the middle. And you can see Tom standing more to the right. And at this time, Dieter was not a member of the group. So one of my most memorable messages from Gobind was one Saturday morning where it's written, 9 a.m., I was here, where were you? So the first rule was you had to show up and be in the lab 24/7. Everyone drank hard. What that meant was during the week, Josephine's coffee. I actually never drank coffee before I went to Gobind's lab, and I rarely drink coffee since because that coffee was really toxic. Play hard. Our group would go to the Muddy Charles and have beer sessions with kids and dogs and go to Vermont on ski trips, and mostly great colleagues. You become part of this Khorana family, and because I always called Gobind in the original LinkedIn, he would always talk about previous people in the lab. You always felt you knew everyone who came before you, and everyone who came after you always thought that they knew you. And that ended, in fact, in many symposia with bringing the whole group together. So how has my experience with Gobind influenced my scientific career? A lot of my career has been focused on cancer, which is a very complex disease. It's a heterogeneous disease. Most cancers have complicated origins. And the dissemination of tumor cells, particularly for solid tumors throughout the body by metastasis, really is the primary cause of death. So what do we know about metastasis? It's characterized by many distinct biological states, but basically the main characteristic of it is stress in the environment. If a tumor at the primary site is fat and happy, it's not going to move. But if it's losing oxygen, losing nutrients, this drives it to become reprogrammed largely in response to its microenvironment and move not only through connective tissue, but actually to invade into blood vessels and lymph vessels and move after extravasation from those vessels into tissues in other parts of the body, where those tumors then grow, take over those tissues, and this is what leads us to death. I would argue that this heterogeneity meaning that we can't kill all cancer cells unless we actually kill normal cells, because there's such a different collection of cells, each of which has a different specialty, some migration, some growth. In order to get them all, because cancer cells are derived from normal cells, we can't avoid killing normal cells as well. So why is cancer so hard? It's a balance between trying to target the cancer cells, but leave the normal cells alone. And so how do we address that? We either need to selectively kill these malignant, meaning metastatic cancer cells, which is really what a lot of people have focused on. Or in my lab, what we focused on is trying to reprogram cancer cells to eliminate the metastatic disease, reduce heterogeneity among the single cells in a population that drive the metastatic disease, and then more effectively kill these non-metastatic cells. So basically, my lab, we've really focused in the last 15 or so years on 2 questions. How can we regulate metastasis? And we have identified both suppressors of metastasis and promoters of metastasis? And more specifically, can we increase drug sensitivity by suppressing metastasis and associated cellular heterogeneity? And the answer to that last question is yes, we have been able to do this, and we're still working on this using different kinds of approaches, targeting different genes, looking at how we can reduce heterogeneity in multiple ways. I decided that instead of going through all the work that we've done in this area, since we're all emerging from a pandemic that frankly has not let go of us, I just had COVID myself 2 weeks ago, that I would talk instead about more recent work that we've done during the past couple of years to try to address the pandemic. And so next slide is a pivot for us to look at COVID. And in particular, what I want to tell you about is a story that we published this year about cannabidiol, or CBD, one of the compounds from cannabis plants, and SARS-CoV-2. As I mentioned in cancer, what is driving metastasis is stress. And so a number of colleagues and I, in the end, about 30, got together and decided to target the early host stress response, which is not only activated by many pathologies like cancer, but also viruses. So the question was, could we use what we knew about cancer and apply it to this new disease, basically knowing nothing about the virus that we were focusing on? And this is just a schematic showing that SARS-CoV-2, the virus that causes COVID, binds to a receptor on the cell surface, ACE2 receptor, and in doing so, internalizes, and the virus, which is an mRNA virus, basically gets translated, gets replicated, gets packaged, and then buds from the cell surface. And so really, all we have, in a sense, as a weapon is either to neutralize it before it gets into the cell, which is the point of vaccines, or to try to immobilize the host immune response, which is also a very important factor in cancer. So I don't want to go through this in detail. This is actually a very early slide, and we've changed some of our approaches for addressing it. But the point I want to make here is there's really two phases that cause either severe disease or death from COVID. The first is from viral toxicity itself. It's the initial viral response phase. The second is kind of interesting. It comes from an overactive immune response that we have. It's our host inflammatory immune response, essentially like an autoimmune response, and it's driven by cytokine storms that we produce in response to the virus. And both of these, particularly, we could see that in the early wave of the Washington strain, caused death. Even when you thought you were recovering from the virus, the second phase was also really important. So when we were looking at this, the host response, we were looking at both, but kind of thinking about that second immune response. So as it turned out, we set up a screen, and I'll show you that screen in a minute. And what we did was basically open it to anybody who had any kind of a compound that they wanted to try to see whether we could suppress in some fashion the replication of SARS-CoV-2. And so this was a team effort. And it turned out in my lab, we tried a few drugs, but one of them, which we tried for a funny reason, and so my story is a lot also about serendipity. We tried it because we were studying a protein called BUC1 in cancer that supposedly was inhibited by this drug, cannabidiol. It had been reported, but we found out it was not. So we had it and we thought, let's try it. What is cannabidiol? It's a natural compound derived from the cannabis plant. And so that plant, sometimes called marijuana, sometimes called hemp. The difference between those plants is that hemp has less than 0.3% THC, which is the psychoactive component in marijuana. As it turns out, CBD in the US is an FDA-approved drug. It's prescribed for pediatric epilepsy. It's called Epidiolex. It's an oral solution. And the key issue is how it gets delivered because it's very hydrophobic, hard to get a lot into the blood, and readily metabolized. But what's nice about it is that in theory, it has multiple means of ingestion, including inhalation, and no need for injection in hospital settings. So as I mentioned, we set up this drug discovery pipeline. And so we tried all these compounds. And in some cases, we used other coronaviruses like OC43, which happens to be a cold virus, and also a murine hepatitis virus. Interestingly, we didn't do that for CBD. We just happened to hand it to the facility looking at SARS-CoV-2 because we thought we would focus on an immune response. As it turns out, it actually doesn't work on OC43 or the murine hepatitis virus, but it worked extremely well with SARS-CoV-2. So we lucked out there. Using this screen, we found lead compounds, we validated them, and then in our case, we did transcriptome profiling, we checked them in animals, and finally, we analyzed clinical data. And so I just want to summarize this work that we did. So first question is actually, why test CBD? And there were several reasons that we thought it might work. One is it induces heme oxygenase, and as it turns out, that is an inducer of interferons and is a general antiviral agent. We later found out that this was not the mechanism by which it works, but as I'll tell you, interferons actually are involved, though obviously activated a different way. It's known to promote a host inflammatory response, which is why we were particularly interested. And it was reported to target expression of ACE2, the receptor for SARS-CoV-2, and protease that's needed for entry of the virus into some cells. So we thought, okay, what happens? First question, does CBD inhibit SARS-CoV-2 replication? As you can see on the next slide, it actually is a potent inhibitor when we looked in lung cells, and these were lung cancer cells. Now, what's interesting is that CBD can come from a number of sources. You can actually isolate it from hemp plants, or you can synthesize it. And because we were worried about some potential contaminants and byproducts, we got it from multiple different sources, including commercially online. And we also analyzed it by using quantitative NMR to make sure that really we were looking at fairly pure CBD. And so we focused on CBD that was greater than 98% pure. And as you can see, this is a viral replication assay where we were looking at viral spike protein in cells. It was very effective at inhibiting SARS-CoV-2 replication. And the EC50 was just around 1 micromolar, which is a bit high for a drug, but as it turns out, is in the right range for the ability of CBD to get into the blood and act, for instance, as an anti-epileptic agent. We also looked at different variants of SARS-CoV-2, and this just shows you the Washington strain, the alpha, beta, gamma. Since then, we've done Delta and Omicron, and it does work for all variants of SARS-CoV-2. Next slide. So what about other analogs of CBD? What about other cannabinoid products? Do they work? That's actually very interesting and shown on the next slide. We tried a number of them that we isolated ourselves or purchased, and some of them were precursors. Some of them varied by just a couple carbons on the aliphatic tail. And as you can see, if you look at the red line on the lower right, that's CBD. There is no other compound that we tried that had the same ability at the same efficacy to inhibit SARS-CoV-2. We were particularly interested in THC. As I mentioned, that's the psychoactive compound that's found in marijuana. That's the one that gives you a high. And so immediately you want to say, hey, I can smoke marijuana and I can cure SARS-CoV-2. As it turns out, that's not the case. THC by itself, as you see on the top, has almost no activity. And that green line just below it is a case where we combine CBD and THC. And not only did THC not work, it antagonized the ability of CBD to work. So bottom line of that one, no, don't smoke weed, marijuana for the purpose of trying to suppress COVID. You can smoke it, obviously, for other reasons. So when does CBD act to inhibit the virus? So as we all know, the vaccine blocks the ability of the virus to enter. And it had been reported that CBD suppresses the receptor, which would also block the ability of the virus to enter. But as we showed, looking at that receptor on the left, CBD has no effect on it, particularly in cases where we overexpress the receptor in cells. But on the right, what we did was add CBD, and we found that it was still effective at suppression completely at 6 hours after infection, and even partially at 15 hours after infection. So it's an early response, but it actually doesn't prevent the virus from entering the cell. So it really is a host response. So what is the mechanism? We're still working on this, but what we first did was look at RNA-seq analysis of what's happening, first of all, with mRNAs from the virus. And what's shown here on the left is a heat map where red means high expression, blue is low expression. And if you look at cells untreated, of course, there are no viral products. If you put the virus in, you see a lot of viral mRNA. If you look at CBD alone, obviously no viral mRNA. But interestingly, CBD plus the virus together, and this was CBD pretreated for 24 hours, causes essentially complete suppression of expression of viral mRNA. And you can see that on the right for the spike protein, mRNA that drives spike protein, and mRNA involved in the nucleocapsid. So what we showed eventually is that the host stress response, in fact, was quite involved. CBD activates it, and in particular, it activates interferons. So what are interferons? Interferons, as shown on the right, trigger an antiviral immune response. In fact, they were even used early on as part of treatment for patients. They bind to receptors. There are different types of interferons. They work through a signal transduction pathway involving STATs, and as well as there are other mechanisms by which they can work, such as using kinases like J and K, not just the JAK-STAT pathway, which point they bind to specific targets and they make downstream products of this interferon pathway that act in various ways to block viral replication. And SARS-CoV-2 itself had been known to reduce, just to a limited extent, the host interferon response. So it didn't suppress us completely, but it certainly was not triggering a large interferon response. By contrast, CBD actually did trigger a large interferon response. So again, this is a heat map, and I'm going to draw your attention to the set, which is 3 from the left. So obviously nothing blue, nothing gets induced. The virus itself, as I said, doesn't really give a good interferon response. CBD does give a partial interferon response, and it's even better when you have the virus present. So we get a very robust interferon response. And when we blocked interferon with antibodies, which is shown on the right, so now we're looking at the viral titer, and the third bar from the left shows that CBD can suppress the viral titer. As I told you, you can get a partial rescue by blocking interferons in the media. So we know that interferons are part of the mechanism by which CBD acts. Interestingly, we also looked at cytokines, and we found that CBD essentially reduces almost completely the cytokine response. So as you can see on the left, you see almost no cytokines in a cell that's not really activated, huge induction of cytokines that leads to that COVID cytokine storm I told you about with the virus. And then whether it's CBD alone or CBD with the virus, you're almost completely suppressing that response. And interestingly, this works with CBD, but not with CBDV, which basically looks exactly like CBD, only it's just missing 2 carbons in the hydrophobic chain. So this argues that it's actually a very specific response, probably a receptor-mediated response, and not just because CBD is a hydrophobic molecule. And this is something that we're still looking at now. What is the target of CBD leading to this response? Well, all of this is well and good, but as many of you may know, just showing something in culture is not enough. And in fact, this led us to looking at chloroquine and even ivermectin, which is an antiparasitic drug that really took off in South Africa when people were very desperate. And in fact, initial reports that it suppressed replication of the virus were even retracted. So we were concerned that CBD might look like these other drugs because we had just done in vitro studies. And in fact, so were the editors of our paper that we eventually published. And at one point they told us without mouse studies, we could just publish it, but we couldn't make any claims. So it's very important to show that it works in mice. And this slide shows you that in fact, it worked beautifully in mice. In this case, we pre-incubated the mice. We basically pre-treated them with CBD for a week. And we looked at the viral titer in lung, which is the lower left, and in the nasal turbinate after we had infected with the mouse and with the virus. And we also looked at mouse weight. And as you can see, in a dose response fashion, we suppressed the viral titer in lung with CBD pretreatment, and we suppressed the viral titer even in the nose, which is very important, particularly for Omicron, which does not reach the lung as much as previous strains, but really tends to stay in the upper respiratory tract. We have since actually shown the same thing, treating mice after infection on the first day, not just before. So we think that CBD has the potential as a prophylactic, but also in early response to getting the virus. But the real question, and the question that we're still dealing with, is does CBD lower incidence of SARS-CoV-2 infection in patients? And so in the next slide, what we did, because we couldn't really do a clinical trial initially, is analyze patients who had been taking this drug Epidiolex and see whether those who have been taking Epidiolex, when they were tested for positive, basically having SARS-CoV-2, so positive tests, whether they were less likely to be positive than the equivalent patients who were not on Epidiolex. And to do this, we had to start with over 5 million patients and use medical records at multiple institutions across the United States and just look at patients who had been tested and try to first break it down to those who were patients who basically had seizures, which is how Epidiolex was used, and then those who had seizures, had Epidiolex, and were matched with, for age, sex, and any other kind of parameter that we could find, with other patients that were not on Epidiolex, but still had seizures, still had kind of same history, and so on. And that took us down to over 1,000 patients. So we started out at 5 million, ended up with over 1,000. But what was amazing is even with this group of patients, when we looked at them, we saw a reduction in the incidence of SARS-CoV-2 positivity of about 50%, which is actually pretty significant. So to summarize what we've found, CBD and its metabolites, but not congeneric cannabinoids. I didn't show you about the metabolites, but at least one of them works. Potently blocks SARS-CoV-2 replication in lung epithelial cells. And CBD acts after cellular infection, inhibiting viral gene expression and upregulating the unfolded protein response and interferon antiviral signaling pathways. Pretreatment with CBD inhibits viral titer in the lungs and nasal turbinates of SARS-CoV-2 infected mice. And a cohort of human patients previously taking CBD had significantly lower SARS-CoV-2 infection incidence relative to matched pairs or the general population. So where does this leave us? What is the general perspective, the takeaway of this? To date, we believe that CBD is the only well-founded cannabis drug candidate for a clinical trial, and we are in the process of trying to set up for a clinical trial. I will say that I contacted Partha in the middle of all this work, asking whether physicians in India or clinicians in India might be interested in a clinical trial, and realized that you were all completely inundated and not ready to try one at that time, at least. So why do I say that it's the only well-founded cannabis drug candidate? As I mentioned, it does inhibit replication of SARS-CoV-2 in cultured cells, and so do a number of other compounds, particularly as noted at the bottom. There was a paper by Van Bremen in the Journal of Natural Products also this year, arguing that some of the other compounds, some of which we tried and found not to be very effective, would work in culture. And that's because he was using about 10 times the concentration and blocking the ability of the virus to enter. But what you have to think about is whether the dose that you're using is comparable to the levels of the drug you'll find in patient blood with appropriate oral formulation. And we know that CBD can be found at the needed dose because it was tested for Epidiolex. And so we know its blood levels, and they are about that range. More importantly, we show that CBD decreases the titer in mice. And this is the gold standard for any potential drug candidate to treat COVID. We also found that it had a lower incidence of positive COVID tests, but we know that this is not sufficient. So we know that we still need to do a clinical trial to test this, but it certainly is very strong supportive evidence. And we made sure that we only used FDA-approved, doctor-prescribed oral formulations of CBD, so we knew what was happening with it. And finally, it's more viable than other polar carboxylic precursors like CBDA, as reported in the Van Bremen paper, which have poor stability and poor pharmacokinetic prospects. So where does this leave us? As I pointed out, we do need carefully designed placebo-controlled clinical trials with known concentrations and high characterized formulations. Perhaps we're not dying at the same rate from Omicron that we did from Delta and other variants, but as we all know, this pandemic, this virus is still with us, not likely to go away, and it may be that drugs can prevent in a prophylactic way or ameliorate the symptoms that we get when we have COVID and make it basically a disease like flu that we can just put behind us. We do need to find the in vivo concentration and optimal route and formulation, but we believe that we do know this based on trials with Epidiolex and with other formulations that we've been working with. And finally, kind of a shock for me, we need more basic science discovery on CBD targets, mechanism of action, as an antiviral, and structural function relationships to identify other potent cannabinoids while ruling out ineffective analogs. And frankly, this is a whole field that if I was younger, perhaps I might jump into because there's so little known about it. And finally, what I would like to say is, as I mentioned in the beginning, perseverance is a characteristic of Gobind. It was a characteristic of my thesis project. And it certainly was a characteristic of this project. We had to do the mice 6 times. We nearly couldn't get this published, but in the end, we were proud to get to the point where we did. And in the final slide, this was not just my group, which is in the upper left, but it was a team effort, as I pointed out, of over 30 investigators, including experts in epidemiology, like Meltzer's group that did the human data, and the lab at Argonne National Lab, run by Glenn Randall, who did all the viral studies, and Guido Pauli's lab at UIC, who really did all the analysis in the pharmacology, and a group at University of Kentucky that did the animal studies, and so many people who contributed to this. And I will say that one of the major lessons I learned from the pandemic is working together can get you so much further in so much less time. And I hope that this will be the future of science as we move forward. And Gobind in his own lab really was a reflection of that. And so with that, I'd like to thank you and open the floor to any questions that might be of interest.
Thank you very much, Marsha. That was a fascinating talk on SARS-CoV-2. Indeed, very fascinating. There is one question from, well, there are several as a matter of, no, there's one question from Professor Umesh Vashni who asks, have you tried it for other viral infections, particularly your analysis with human population taking CBD?
So we have not done any analyses, particularly with human populations for other viral infections. And frankly, even within the coronavirus group, we believe it will work for SARS-CoV-1 and SARS-CoV-2, but as I mentioned, it doesn't even work for OC43. So we're not arguing that we were hoping that this would be a general antiviral drug that, you could think about for any virus that comes through. But I think it really likes SARS-CoV-2. That seems to be, or the SARS lineage. So other viruses remain to be explored.
There's a laudatory comment that says, great talk, and a question, can you say a bit more about potential molecular targets?
Yes, I can say a little bit more about that. The only targets that are known are actually the so-called cannabis receptors, which are actually targets for THC. And it does seem there's a bit of a yin-yang for how THC works and how CBD works. And it's known that CBD does not bind these receptors particularly well by itself, but if THC is bound, then CBD can modulate that binding. And when I say there's a yin-yang, the best story, after I got into this, I met with a lot of researchers in the field, and maybe the best story was I met some chickens who had been on CBD and THC, and it turns out when they're on CBD, they're very lethargic and they lose a lot of weight. So you can counteract that by giving them THC, and just like those of you who've ingested pot in different ways, you get hyperactive and have an amazing appetite. So we do know that there is a kind of yin-yang between these different compounds. We do know the receptors for THC, and we are very actively looking at what the receptors are for CBD. The other thing that's known is they do affect ion channels, which is not surprising, particularly for the epileptic kind of responses. Whether they are direct targets or indirect targets, I think this stage is not entirely known. So you can find proteins that do respond, people call them targets, but direct targets are not known.
So there's a follow-up question, Marsha. The follow-up question is, what about orphan GPCRs?
Orphan GPCRs?
Right.
I'm not sure about the context of that.
Possibly as the target. Possibly as the target.
Oh, as the target. We have not personally seen any evidence for that. We are trying several approaches involving proteomics, involving cross-linking, and so on. So it's a possibility, and we do obviously have endocannabinoids. So our body has versions of these. Otherwise, we obviously wouldn't be responding to them. But at this stage, I really do feel a lot more work needs to be done to understand how these drugs work.
So one last question. Why did you look for the effectiveness of cannabis derivatives in particular?
So I mentioned that briefly. This is, again, the serendipity aspect where we were looking, we thought that we would follow HMOX1, which can lead to interference in an antiviral mechanism, because this is one of the mechanisms that has been used for fighting viruses. And we were working on a protein that antagonizes HMOX1. So we were trying to inhibit this protein, and it was reported this protein is called BOC1. It's reported that CBD inhibits this protein. So we thought, oh, maybe that's a way of activating this. And then we did our own studies, and it just tells you don't believe every report that you read. CBD activated HMOX1, but it actually did not inhibit BOC1. But then we found out that HMOX1 was actually not the mechanism for the interference anyway. So we tried it after all that, just saying, hey, we have it, affects the immune response. Let's forget about viral replication. Let's just look at the lysates. That's why we skipped OC43 and the other viruses. Let's just look at the lysates and see what it does to cytokines. And to our shock, it actually blocked the ability of the virus to replicate. So as I said, this is a story of serendipity. Can't even give myself credit for thinking that it would work in this fashion. And also perseverance, because there were many times when we thought, okay, we'll never get these mice to work. And, we'll never get beyond step one. But the one thing I do want to stress is in the course of our studies, we found that the quality control, not sure about India, but I'm guessing it's similar, is very poor for CBD. That a lot of the CBD that you would buy online is synthetic, which is actually, frankly, the best CBD, much better than trying to isolate it from hemp. But often it's stored in a poor way, and you have, as a consumer, no way of knowing that it's good. Plus, it's very hydrophobic, so if you just took drops or powder, it wouldn't really work. You really have to take it in a special way such that it gets into the blood at the level that's needed. And so the formulation and the concentration is super important. So after this finding, as you can imagine, particularly with this other paper that came out, this story went viral. Talk about YouTube. Lots of press were involved and so on. And I found myself saying, essentially, here's our story, but please do not go to the dispensary, buy CBD, take it, and so on. Wait for our clinical trial. And I still say that. We hope to get it done soon, and hopefully it will be positive, and we'll be able to tell everybody how best to take this. And with luck, it will have an effect on this disease that we're still confronting.